Hello, everyone. This is my review video for AP Biology, Chapter 16, the molecular basis of inheritance. So this is basically going to cover the structure of DNA and RNA, what makes up DNA and RNA, the differences between the two, and then the process of DNA replication. So let's just get started. So first we have the structure of DNA, right? So DNA is just made of three-part monomers called nucleotides. And just remember that monomers make up polymers, and those polymers are made through dehydration and synthesis, which is basically the removal of a water group, which allows for a covalent bond to form, which is called a glycosidic linkage, if I remember correctly. So the three parts to a nucleotide, you have the nitrogenous containing base. Nitrogen is super important when it comes to nucleic acids, especially, and proteins. But you have the nitrogenous containing bases like adenine, thymine, cytosine, and guanine, and uracil for RNA. You have a deoxyribose sugar, which is just a sugar, which has 6 carbons on each corner of the sugar. And then you have the phosphate functional group. So that's just a PO minus 3 phosphate and 3 negatively charged oxygen. And that makes up a phosphate functional group. And then DNA has a double helix shape, which is pretty common knowledge. So this image up here where a mouse is basically just shows the structure of nucleotide. You have the PO minus 3, the red highlighted phosphate group, have the deoxyribose sugar. So a super important thing to remember is how the carbons are organized in any sugar. So carbon 1 is the one that's directly right, or directly, okay, that's the wrong word, sorry, directly clockwise from the oxygen. And you obviously have the hydroxyl groups. And then here you have the nitrogenous containing base, which you don't know what it is in this case, but you do know it's a purine. I'll get to that in the next line. And down here you can see the double helix shape. It's a nice visual, right? That. Okay, the more structure stuff. So obviously you have the base pairing rule, only certain bases bind to each other. So adenine binds to thymine, and cytosine binds to guanine. I know that for some reason everyone likes to remember it. Cytosine binds to guanine, but it's actually going to make it easier to remember the purines and pyrimidines if you remember guanine binds to cytosine. And I'll show you why in just a second. So purines, they're basically the two bases that are double rings. So you have adenine and guanine, while pyrimidines are single rings, which is cytosine and thymine. If you ask me, the easiest way to remember that is conceptually. So the important concept to remember is that when you're pairing two bases together, only a purine can bind to a pyrimidine. So if you have two purines that bind together, if you have two double rings bases, then that's going to take up too much space, and the DNA isn't going to be evenly aligned down the center. But then if you have two pyrimidines that bind to each other, so two single rings bases, then you're going to take up too little space, and then spacing is also not going to work. So basically you won't have an actual DNA molecule if you bind two purines or two pyrimidines. So the way I organized it here with ATGC, so adenine, thymine, guanine, cytosine, the first two are going to be the purines. So adenine is the first one in the adenine thymine pair. Guanine is the first one on cytosine pair. First ones of adenine and guanine are going to the purines, and thymine and cytosine, the second ones, are going to be the pyrimidines. So yeah, that's actually 2 ways to remember it. Either you can know the conceptual stuff, so the purine has to bind to pyrimidine, otherwise it's going to take up too little or too much space, or just memorize it by the first one is purines by AG, second ones are pyrimidines by PC. And then you have the actual bonding within the DNA molecule. So when you pair the bases together, they are going to form hydrogen bonds, which is most commonly going to form between hydrogen oxygen or hydrogen nitrogen, I believe. So in the case of adenine thymine, they pair with 2 hydrogen bonds, and cytosine guanine, they pair with 3 hydrogen bonds. Easy way to remember this is just they're the same order as the actual base pairing rules. So adenine thymine, cytosine guanine, they're the same order. So just 2 plus 1 equals 3. That's pretty easy to remember. I think the conceptual as to why it's 2 versus 3 is just outside the scope of this class. And then in the actual backbone of the DNA, where the phosphate binds to deoxyribose sugar, there's going to be covalent bonds between that phosphate group on one nucleotide and that deoxyribose sugar on the other nucleotide. So as you can see up here, you have this red rectangle, which that just indicates one nucleotide, roughly. So the yellow backbone, that's just going to be approximately where the phosphate group and deoxyribose sugar is. And then the purple green indicates a hydrogen bond adenine thymine. Down here, you can see the 3 proteins, so AG, GC, and uracil. Function of DNA is there's a whole host of things, but the main one stores genetic information. So it stores and what that means, stores genetic information, it contains genes, which are used to determine many observable phenotypes, like your eye color, your hair color, for example. It codes for pretty much almost all the proteins in the body through process and transcription and translation to cover in the field. It does many other important things, and sometimes harmful things if like bacteria invades or virus invades. They can take control of your cell too much, which is obviously a bad thing. So the structure of RNA, I'm not going to go into nearly as much depth as I did with DNA, since it's almost the exact same, except the only difference is that instead of double strand, this is a single strand molecule. Instead of thymine, it has uracil, so uracil replaces thymine, and instead of deoxyribose sugar, it contains a ribose sugar. RNA's primary function, there's many more secondary functions that probably we haven't covered in this class, but primary function is to assist with ribosome building through ribosomal RNA, amino acid carrying through tRNA, and carrying the actual sequence of complementary base pairs that code for proteins through codons. That's going to be mRNA, messenger RNA. Okay, so now we're on to the next main concept of DNA replication. So I think the most important thing of DNA replication is understanding the actual flow of replication. I know usually when we cover this stuff, we're more focused on like enzymes and their functions, but it's really, really important to the entire flow, like the big picture stuff. Although once you know that, the enzymes are going to make a lot more sense. So just to put things in perspective, each cell in your body is going to have an identical copy of DNA, or at least almost identical RNA. Within each of those cells, when they divide, DNA replication is going to happen in the S phase and synthesis phase of cancer. And when that replication happens in each cell, there's going to be this arbitrary point called the origin of replication. I don't remember how that origin is identified as a starting point. That's a minor detail, it's not too important. But let's just say that you have an origin of replication at this point. The replication happens in both directions. So the right of that origin and the left of the origin. Replication is happening is happening simultaneously, which obviously makes the whole process super streamlined, super efficient, but also makes it more confusing. But this picture is actually really helpful, helped me understand it pretty well. So from the origin, it goes left and right. And since replication is happening on both sides are going to have a leading strand and a lagging. So to review what those terms mean, leading strand is pretty much where 5' to 3' end points away from the replication fork, or sorry, towards the replication fork. And the lagging strand is where 5' to 3' end points away from the replication fork. Replication, and obviously the replication fork is where the unzipped DNA intersects zipped DNA, at least vaguely speaking. By unzipped, I mean the helicase hasn't acted upon DNA yet. So that should better help visualize the entire process. It's happening two directions at once simultaneously. Each side has a leading and lagging strand. All the enzymes are acting on both sides simultaneously. So let's go specifically into those enzymes now. So first enzyme, we have topoisomerase. So as we covered in the previous slide, or DNA is double-stranded. So topoisomerase pretty much flattens that double-strandedness into what you see in this image, like a two-dimensional, well, it's not two-dimensional, but like this flattened version of the DNA, which makes it possible for the next enzyme, helicase, to unwind the DNA to two separate strands so that the enzymes can work easily on both of the strands instead of trying to work when it isn't unzipped. Is actually a possible thing. And then the numbers on the actual image just show you like a sample of where that enzyme would work. For number 3, single-stranded binding proteins, the number isn't fully representative of all the areas where those proteins would act. And they would be pretty much on both sides on leading and lagging strands both. But yeah, the single-stranded binding proteins basically hold apart the two strands of DNA so they don't drift apart or reattach. They stay in place pretty much. Continuing on with the enzymes, so you have RNA primase, which is going to place the RNA primers on the leading strand. That primer, there's only going to be one primer on the strand. It's going to be completely continuous because most of the enzymes in DNA replication are very friendly on the leading strand, so 5' to 3' towards the replication fork. All the enzymes work very well, very efficiently with the leading strand. Then you have the lagging strand, which most of the enzymes are, they don't really work well. They're kind of hostile to each other. So RNA primer, or the RNA primase, has to place a bunch of different RNA primers instead of just one continuous RNA primer. Makes things a little bit more complicated. Then you have DNA polymerase 3, which synthesizes a new strand of DNA just by attaching the complementary base pair. That's pretty straightforward. DNA polymerase 1 then hops in behind, behind is the right word. Behind DNA polymerase 3. It basically removes the RNA primers and replaces those primers with DNA nucleotides. Really, the only thing you're replacing is thymine and uracil. Those are the two things that are differentiable between the RNA primers and nucleotides, or DNA bases, sorry. Just to make clear the location of all these enzymes, all three of these enzymes are across the entire length of the origin fork of replication, happening at both ends of the origin of replication on both leading and the lagging strands. More enzymes, how fun. Okay, so on the lagging strand, you have ligase, which is basically going to connect all of the Okazaki fragments together. The Okazaki fragments are pretty much what all those RNA primers, those separate RNA primers, were turned into after they were synthesized into complementary base pairs by DNA polymerases 1 and 3. Now they're Okazaki fragments, but they're still not attached together. So ligase basically connects the Okazaki fragments and then gets rid of the empty space. By empty space, I mean no fragments there. And then DNA polymerase 3, and I think there's another enzyme that does this. Well, I don't remember what that name is. The DNA polymerase 3 basically repairs a lot of the errors that are made in replication by going back and correcting those errors. The new DNA molecule that is made is semi-conservative, meaning one strand is from the previous DNA molecule before it's replicated, and one is synthesized. So that's pretty simple to understand. Yeah, so that is the entire slide. This is only, I believe, a third of sickle cell units, but tune in next time and I will cover protein synthesis and exons, introns, alternative splicing, spliceosome, the post protein synthesis activities or post transcription activities. And then after that, we're pretty much going to be a wrap on sickle cell units. So thanks for tuning in. Thanks for listening. See you next time.